Okay, in this last video for the species module, we will continue to build on those techniques of barcoding and metabarcoding that we introduced previously, but we'll talk about one specific application, eDNA, that is, again, something that's relatively new in the field and a really exciting development that opens up some new possibilities for what we can use molecular ecology for. So we've introduced eDNA briefly previously, but just to recap, environmental DNA, are often abbreviated as eDNA, is a mixture of DNA that originated from the organisms that are in that particular environment. So in this example, what I'm showing is one application they've actually used eDNA for in Southern California. They've actually used eDNA to monitor whether great white sharks are present in the water or not, in hopes of being able to prevent interactions with humans. So the idea is that if we're talking about the ocean environment, any organism in the ocean, so like a great white shark or all the other fish species that are there, what they're doing is they're releasing cells and they're pooping, so that's shown here, and that releases DNA into the environment, or eDNA, and then you can collect a water sample that contains some of that DNA that's been sloughed off from all of the organisms that are present in that habitat or that community. So eDNA is one way for us to molecularly estimate what species are present in that community without having to physically see or sample the organism itself. And so oftentimes when we're dealing with eDNA, there are two different possible types of samples we can get to estimate what species are present in that community. We can either get water samples, so that can be from the ocean, a stream, a lake, or soil samples. So soil is another common way to obtain eDNA. In both of these cases, what we're doing is collecting a bulk sample. So we've talked about bulk sampling before. That just means we're taking a sample that contains the molecular information, the DNA, from many different species at once. And eDNA is useful for a couple different purposes. eDNA, like I said, is a way for you to estimate what species are present in that community without having to actually sample or see that organism. So that can be useful for a couple different applications, one of which is monitoring invasive species. So in this picture, what I'm showing here is an invasive quagga mussel, which is actually a problem in many California waters. There are systems set up throughout California to test our water system to make sure we don't have quagga mussels. So in this picture, obviously, there are lots of quagga mussels. So if we did a visual survey, we would be able to tell if they were there. But really what we want to do is catch it before it gets to that point, right? So with eDNA, even if there are only a few quagga mussels present, we'd be able to detect them with eDNA and hopefully prevent this scenario from happening. eDNA is also really useful for monitoring endangered species. So something that maybe you might not want to disturb by taking a physical sample from them, or they're hard to find because there aren't that many of them in the wild. So one example of an eDNA study that's been done, many have been done, but one of the ones mentioned in your textbook is in Borneo. What they did was monitor for endangered mammal species, including the Asian elephant. This is actually a picture I took in Thailand. This is, I think, one of my favorite pictures I've ever taken. If you notice there's some motion happening in the ear region, that's because as I took this picture, that elephant smacked me in the face. But endangered species are one good application for eDNA. It's non-invasive, meaning we're just taking a sample from the environment and not having to actually disturb or harm the animal at all. So here's an image of what the overall workflow would look like if you were doing eDNA analysis using water samples. So what you would do first is collect your water samples from a stream or a lake. you would filter those water samples, and what that filtering process would do is collect all of the eDNA on a filter. So what we're interested in is not the water itself, but all of that environmental DNA that's been sloughed off from the sharks or whatever is in that environment. So we can collect all of that eDNA on a filter and then extract that eDNA from the filter. And from that point, we can take two different general types of approaches with our eDNA analysis. On the left here, we can do what's called a species-specific assay. So this is the route we would take if we were interested in whether one particular species was there. So that example I talked about earlier with the great white shark. In that example, that was the main thing that researchers were interested in, is whether or not that particular species was there. So in that case, they would want to use a species-specific assay, and the way that they accomplish that is by using PCR primers that only anneal to that one target species. And then it also mentions in this box that if you just want to know if that target species is there or not, you can use conventional PCR. Remember, the other type of PCR we talked about is qPCR, or quantitative PCR, that also gives you some estimates of amounts of how much of the DNA from that target species was there. So if you use qPCR, you can also roughly quantify how much eDNA is there, and then you can say not only whether that species is there or not, but roughly what the abundance is for that one particular target species. The other approach you can take if you aren't interested in one target species, but rather you want to know overall what species are in that community, in that case, you would take a metabarcoding approach. So remember, as we've talked about before, when you're doing metabarcoding, you're using universal primers that amplify species from a broad taxonomic group. So if you wanted to know not just the great white sharks, but all types of fish that were there, you could then use this metabarcoding approach, in which you would use universal primers and then that high throughput sequencing to get DNA from multiple types of species at once. And just like I've talked about previously with metagenomics, I actually have another master's student in my lab, Jason, who's currently using eDNA techniques for his thesis. So what he has done is collect water samples from different local streams and creeks. And what he is interested in specifically is the timing of juvenile salmon emigration. So basically when those baby salmon are leaving the streams and going back to the ocean. So if he's interested in one particular species, which type of eDNA approach should he be taking? A species-specific approach with qPCR or a metabarcoding approach? What do you think? Right, so if he's interested just in this juvenile salmon, so that's one particular species. So here in this image here, say you're collecting water samples from a community that contains all these different types of fish. If Jason just wants to know if juvenile salmon are present or not, or roughly in what amounts they're present, he would take a species-specific approach with qPCR. If he wanted to know all of the fish that were present in his water sample, in that case, he would take a metabarcoding approach. So this illustrates the differences. In either case, the starting point is the same. You're taking a water sample that contains eDNA, but then depending on what your study question is, you're choosing which approach to take, whether you want to characterize all the species that are there, which would be metabarcoding, or whether you just want to focus on one particular target species like juvenile salmon, that would be your species-specific approach.